Hi guys, now one of the things that you'll need to know for F211 is how to measure lung function, and one of the devices that you can use to do that is the spirometer. Now the diagrams sometimes can make the spirometer look quite complicated. In fact, it's a very simple device. We have one right here. I'm just going to show you basically what it is. Okay, so if you come around here, the person will be breathing in to here. Usually there'll be a nose clip to make sure that the person is just breathing in and out completely through the spirometer and no air is going in and out through their nose. Okay, so we're breathing in and out through here. The air goes in, and if you have a look here, just come a bit closer. Essentially what we have is a water-filled bath with the inlet and the outlet that come from here, all right? So person breathes out into the tube, the air comes in here, and then when they breathe in, the air goes back down the other tube, and so on. Now what happens is, because this is a water-filled area, when we put this on, put the hinges down there, it means that the space inside this container is sealed, airtight seal right there. That means when I breathe in, the volume inside this is going to go down, and when I breathe out, the volume inside it is going to increase. And that's going to essentially pull the lid down when I breathe in and push it up when I breathe out. And I'll show you what I mean, okay? So remember, basically just air inlet and outlet, or gas air inlet and outlet, and simply it's on a hinge so it can move up and down. And if I put this down, now it's completely sealed by the water, okay? So very simply then, I would have a nose clip properly if I was doing it properly, and if I was doing it properly, we would also have pure oxygen inside this chamber here. But I just want to show you how this works, okay? So, you can see this part. Okay, now what I want you to notice is, as I was breathing in and out, it wasn't going all the way back up right to the top. As I was breathing in and out, it was slowly, with each breath in and out, it was getting slightly lower and lower and lower. And we will talk about that in a bit. But I hope you can see that there's nothing very complicated about this device. The only extra thing that's in there is a chamber that's there to absorb the carbon dioxide, and we'll talk about that as well. But essentially, you breathe in, lid goes up. You exhale and the lid goes up. You inhale and the lid goes down. And we'll talk about the modifications of this later on. But essentially, that's a spirometer. Thank you.
All right, guys. So you've just seen how, what a spirometer looks like. So we'll now continue our discussion about how it works and how that device that I just showed you can be used to measure lung function. Okay, so we'll be talking in detail about the spirometer, how it works, and we'll just touch briefly on the kind of questions that might come up. Okay. Let's begin. So the device that I showed you, essentially, remember, was looking at how a person's inhaling and their exhaling gives you a measure of how well their lungs are working. Now, remember, when you inhale, your lungs are expanding, and that reduces the pressure and that draws air in from the environment. And when you are exhaling, a reduction in lung volume causes air to move out. Okay, so we're going to be looking at, essentially it's going to be to do with the volumes of air going in and out. Okay, and that's primarily being measured in decimeters cubed. Okay, we'll also be talking about oxygen and carbon dioxide. Okay, remember, this whole process is essentially linked by the equation for aerobic respiration, which is glucose plus oxygen gives you, with the release of energy in the form of ATP, gives you carbon dioxide plus water. Okay, so what we are thinking about is that when we breathe in, it's oxygen essentially that we are exchanging into the blood so that it can be transported to the tissues, so that the cells of our tissues can carry this process out. And at those tissues, the cells are producing CO2, and this is entering the blood to be exhaled. Okay, so just remember this, we've been through that already, but now we're going to look at how this is measured. Okay, so this is essentially the device that I showed you. It looks complicated, but I hope I can convince you that it really is, there's nothing really much to it. Okay, so this chamber is filled with oxygen. The spirometer chamber is filled with oxygen. With medical grade oxygen. Okay. Okay, so its total volume inside is filled with oxygen, is full with oxygen, and none of that oxygen escapes, because as this dotted line shows, this is the water level, and the chamber goes below that, so it's essentially, it forms an airtight seal. Okay, it forms an airtight seal so that oxygen is not going anywhere unless this person breathes in. Okay, so let's follow the story then, starting with the oxygen in this chamber. So we'll just highlight that oxygen is in here, O2, and what's going to happen next is, as this person inhales, their lungs are expanding, so they are inhaling, and as they do that, the oxygen then is forced into this tube right here. Okay, so a person is inhaling, oxygen moves down the tube, goes through the tube all the way through here, round here, and in. Made it. Okay, and down into the lungs. Okay, and the gas exchange process will happen. Because of the high concentration of oxygen, it will diffuse into the capillaries, and because of the low concentration of carbon dioxide in the lungs, it will move from the capillaries into the lungs. And so here we continue our story with carbon dioxide. So carbon dioxide coming out, or air containing lots of carbon dioxide, comes out and goes down this side. Okay, and it will go all the way around, through here, up through here. We'll talk about this chamber later. And essentially right back through and back into the chamber. Okay, so there's not really much to that. I hope you can see that. As the person breathes in, the chamber filled with oxygen, that chamber will move down. Okay, so let's just make a note. Spirometer chamber, upon inhale, upon the inhale, moves down. Okay, and the amount that it moves down gives us an indication of the volume, but again we'll come back to that later. Okay, so when the person breathes in, the chamber goes down, and when they breathe out, the gas is returned back to the chamber and the chamber goes up. So it's not really much to that. I hope you can see that now. Okay, upon inhale moves down, upon exhale moves up. Simple as. Okay, so here are, I mean, that's the main thing, and it's not much more complicated than that. And these movements are recorded on the kymograph. So I hope you can see here, right here, that this chamber is connected to a pen type device, and that pen is pushed against a kymograph that is rotating, and as it's rotating, the up and down movements of the pen form a trace, like so. Okay, and that is simply that. So the basics of the spirometer is that breathing causes the spirometer chamber to move up and down to, and these movements are recorded by the kymograph, producing a trace. Okay, and it's the trace that you will have seen diagrams of. Some of them, you know, look like that, etc. And we'll come to that. We'll come to what that means. Okay, but essentially that is that. Yeah. Okay, so breathe in, chamber moves down. Breathe out, chamber moves up, and movements are recorded on the kymograph that give us a trace. There are certain things that we will come back to regarding carbon dioxide and O2 consumption and production, but essentially that's how the spirometer works. Okay. The other thing is that there is a nose clip that has to be worn by the person who is using, who is being tested on using the spirometer, simply because you want to make sure that all the gas going in and out of the that's being breathed in and out is being recorded by the chamber. So you don't want to leave the nose open so that inhaling and exhaling could be occurring without it being recorded on the spirometer. Okay, so now that we've discussed the basics, let's move on to looking at what this trace is telling us. Okay. Right, so I did want to just show you some examples of what different spirometers could look like, simply because this diagram's a bit complicated. This one's much simpler. This one is from an exam paper. The spirometer might not always be shown in this way. It could be shown in different ways, but I want you to be aware that essentially what we've spoken about is there in every single one of these diagrams. A water-filled container with a chamber on top. The chamber is linked to the air inlet and outlet so that as the person breathes, it changes the volume of gas in this spirometer chamber, causing the spirometer chamber to move up and down. That movement is linked to the movement of a pen on a kymograph, which produces a trace. Okay? And you can, in this way, you can measure different aspects of a person's lung functions. So let's move on. Okay, so I hope you can appreciate that what we're looking at here, right? So as the person breathes in, the inhale, the chamber loses its volume, and so the trace goes down. And as the person exhales, the gas volume is increasing in the chamber, gas returns to the chamber, and so the chamber goes up. It's not that simple. There are complicating factors, but essentially this is what we are talking about. Okay? So different things you can measure in this way are normal breathing, so tidal volume, the breathing of a person at rest, that's indicated by these peaks and troughs here. Okay, so the volume of air or gas that's exchanged with each breath is indicated by the difference between this mark here and that mark there. Then, if the person breathes in as much as they can, that will indicate their inspiratory reserve volume. Okay, so that's the total volume that can be inspired, okay, or inhaled, right? So that's this measure right here. Okay. Then you can also do the same thing in the opposite direction. So you can breathe. Oh, so what did I say? Yeah. So you can also breathe out as far as you can, and that will be your expiratory reserve volume. That's the extra volume that you can breathe out, not that you do with every single breath, but the volume that you can possibly call on if required to. Okay? So that's the expiratory reserve volume, and that's indicated from here to here. Right? And if you take the maximum to the minimum, so this distance right the way from here all the way down to there, that's the vital capacity. That's the maximum volume of gas that you can exchange, even during exertion. So when your body is going through some kind of physical activity, exercising, maybe on a treadmill or on an exercise bike or something, the person that will be also connected to a spirometer, you could measure how deeply they can breathe and what's the maximum they can exhale during that time. Okay, so that's the vital capacity. Now, even when you breathe out as much as you can, there's still gas left within the airways of your lungs, okay, because they don't collapse completely. You have the C-rings of the cartilage in your trachea, in your bronchioles, preventing your lungs from being flattened completely. Therefore, there will always be a little bit of gas in your lungs, and this volume is called the residual volume. Okay, so even after you exhale as far as you can, there's still gas left in your lungs. So when that is added to your vital capacity, you get the total lung capacity. Okay, now you do need to remember these things, but once you have that down, it will simply be a measure, on the day of the exams. It will simply be, what am I doing here? Anyway, on the day you might be asked, you'll have a scale here, We'll have a scale there, and it will be marked with decimeters cubed here, and you might be asked to measure the tidal volume for example, and you just have to simply read off the graph and then just calculate the difference. That will be the tidal volume, and again for all the other volumes is simply reading back off the graph and getting the difference. Okay, and don't forget your units. That's all there is to that. Okay, now next. Now this is what a real trace looks like, and as you can see, the level does go down over time. If that's slightly confusing, I've also got an example from an exam paper right here. You will notice that with the volume going up, so with each exhale and then inhale and then exhale again and then inhale, you will notice that this graph, this trace, will be going down over time. Okay, so wow, that line is rubbish. You will notice, hopefully this one's going to be better, you will notice that there's a general trend of the trace to go down over time. It's not really as simple as what is suggested here. Okay, so the trace goes down over time. Now why is that? Now let's discuss why that is. So in your textbooks you will read that this line is an indicator of the fact that the trend goes down. So because the trace goes down over time, what this indicates or indicated is either CO2 production or O2 consumption. Okay, now let's look at why that is, and ultimately this comes back to realizing that what's going on here is cellular respiration in the cells. Okay, so let's go back to this. Remember that when you breathe in, you're breathing in the pure oxygen. When you breathe out, you're breathing out carbon dioxide that has been produced as a result of respiration. Okay? Now, let's go back to this. In all the most of the diagrams, you will see this carbon dioxide absorber. Okay? The carbon dioxide absorber. Now, this is usually something called soda lime, and what it does is when the person breathes out, so we're following this blue line again, as the person breathes out, the gas that they've exhaled passes through this carbon dioxide absorber. So the air that leaves the chamber is lacking carbon dioxide there. So any carbon dioxide that the person was breathing out was absorbed here, and so pure oxygen is returned back to the chamber. Okay, so what we should add is that this reacts with exhaled carbon dioxide, returning the remaining returning the remaining oxygen back to the chamber for the person to breathe in pure oxygen again. Okay? So what this does is, because some of the carbon dioxide has been removed, what that means for us and our trace is that when the person was breathing in, okay, they were breathing in pure oxygen, but when they were breathing out, they were breathing out carbon dioxide and oxygen, and because the carbon dioxide, the carbon dioxide reacted in the carbon dioxide absorber, the total volume wasn't returned back to the chamber. Okay, so therefore, when the person breathes in again, they're breathing in pure oxygen, because the carbon dioxide was actually absorbed. So they'll take a deep breath in again, and when they breathe out, the carbon dioxide will be absorbed, so the total volume doesn't go back to the chamber. So the volume exhaled is the total volume of gas exhaled is always slightly lower than the total volume of gas inhaled. What's the difference? The difference is that any CO2 that was exhaled is absorbed by the carbon dioxide absorber. Okay? Now what this means is, if we go back to our equation, if we're losing a certain amount of carbon, if we're losing all the carbon dioxide that's exhaled, that actually gives us an indicator of how much respiration is going on. Okay? Because if we are losing carbon dioxide, that means that that's an indicator of the amount of cellular respiration happening. And according to this equation, actually, the equation should be different. The equation actually should be this. So if we look at the actual numbers of molecules involved, what we should be looking at is at this C6H12O6 being the glucose, plus 6 molecules of oxygen with energy, 6 molecules of water, plus 6 molecules of CO2. Okay? So I hope you can see that the number of molecules of oxygen used up is the same number as the molecules of carbon dioxide produced. So what that means is, if we're getting a certain reduction, if we're losing a certain volume of gas because that's the volume of carbon dioxide being consumed, then if our trace is going down and we're losing that volume because that's the volume of carbon dioxide lost, what that actually also is the same volume of oxygen that was being used up. Okay? So this trend that goes down, if we're saying that the total volume of our chamber is going down because the carbon dioxide is reacting with the carbon dioxide absorber, if this is the volume of carbon dioxide that is lost because it's reacted with the carbon dioxide absorber, then it must be the same volume of oxygen that's being used up when we breathe. Okay? Because the amount of carbon dioxide produced must be the same as the amount of oxygen that is utilized by the cells. Okay? As we can see, the reaction tells us that for every 6 molecules of oxygen that are used up, 6 molecules of carbon dioxide are produced. So if we lose 0.5 decimeters cubed of carbon dioxide over a certain number of minutes, it must mean that we've used up that same volume of oxygen in the same amount of time. Okay, so that trace going down, gives us an idea. So if we divide that X volume of CO2 that's being lost over a certain number of minutes, we can get an idea of a rate of oxygen consumption or usage or utilization. Okay, if we divide the volume by the time, we will get a certain amount, a certain volume of oxygen used per minute. Okay, and that will be a rate of oxygen consumption, or a rate of CO2 produced, carbon dioxide produced, either one. It's indicating the same thing, it's indicating the rate of this reaction happening. Okay, or process. Okay, so I hope that's making sense. I think that covers all the main things. We've talked about how this device works. We've talked about what the different ways are that it could be depicted. We've talked about what the trace means in very basic terms. And we've also talked about what the trace going down over time means. Okay? And I believe that covers spirometry. Alright guys, so if you have any questions, do put them in the comment section. Otherwise, I will say good luck.